So good morning all. Let's start. Our guest speaker is Bruno Camilo. He is expert in histology, bone tissue, and I'm going to have about 2 hours lecture with him. He came especially from Universidade. He's been doing great work on the Society of Natural History in Torres Vedras. So let's welcome him with applause. What is his yours?
Okay, good morning to all. Thank you very much for the invitation. And so I will try to do it in 1 hour and a half, more or less, so we can have some practice. Because I think it's important for you to understand some of the types of visualization of histological samples on a microscope. Therefore, I will not talk about the visualization of samples. And as always,
Se o senhor está ali, lá.
no, talk about that. You worked 3 minutes ago.
Clique no espaço. Clique no teclado. Põe os dois ecrãs.
Já está só.
Não ecrã,
Está melhor assim?
Sim. Meanwhile, we have books to offer and papers to offer. You can take one book home each.
Okay, let's start with the problems. Major problems in paleohistology. Paleohistology is very important and very necessary to understand some important aspects of bone biology. However, you have to think first if you want to study histology or a group. First thing is the question that you wants to be answered. Because no one, or probably a few people, will allow you to cut important bones, because this is a destructive type of analysis. And then when you choose, you have to know to choose which bone I have to sample. Because if you're going to sample a vertebrate, we have no results about the animal. Okay? So I will talk about it later. About the best bones to cut to have maximum information as possible. You have to know how to cut. You have to know, or at least rely that attribute that function to someone that knows how to do it, because you have only one chance. Imagine you go to a museum. I want to cut this bone for my master thesis or for my PhD thesis. Let's say, okay, you can cut. You cut. You go to the lab. Something went wrong. You cannot sample anymore. Okay. And as you know, especially when we are talking about holotypes. Other big problem in paleohistology is that you have to remember we don't work with populations. So if you cut a bone from one species, The information that we'll have is only about the growth record of that bone in particular. Okay, because not all the bones grow at the same rate. There is isometric growth, very rare. There is mainly allometric growth. For some of you that want to get in Paleohistology, or at least to know a little bit more, there is some books, it's not updated, there is 2 more books, very good ones, that can help you to better understand bone biology and the problematics and the potential and the limitations of histology on the study of fossils. So, basically, histology is a very brief definition. We are studying the mineralized tissues of fossil animals. We can talk about eggshells or eggs are not bones, they are different type of material, but we can, through the techniques of histology, we can take some information. But what we'll see is completely different from, of course, a bone or a teeth or even an osteoderm. Historically, histology started almost at the same time as the study of dinosaurs began. However, the interest on histology and on the fossilized bone, the problematics that they have were very limited, and also the resources to study it. At the time, they have basic microscopes. You can see something, but you cannot see in detail, for example, the organization of the fibrillar matrix of the collagen in the bone. They can see osteocytes. They probably could see some types of secondary remodeling, but the meaning of it was just its presence. You don't know the mechanics, you don't know the potential of it. This changes on the 60s, last century, with the works of, mainly from the French teams, by Armand Ricolet, Jacques Taquier, and many others that start to look at the bones in a different way. And then after there is a stop, I would say, in the 80s. And then on the 19th, we have like a boom on histology work. So we have 3 main signals that we can see on on the microstructure of fossil bones. You can see ontogeny. Okay, you know what ontogeny is? The growth process and the time and aging of an animal. You can see phylogeny. This one is tricky. You cannot say, if you see an histological sample, you will not see any specific from that species. You can say this animal grew like this, it died at this age, blah blah blah. But you cannot say this is typical from this species. But however, at some point, there is a phylogenetical sign. Because if I look for histology of ornithopod bone, I say this is the model of growth of ornithopods. Or for a pterosaur, this is the model from not only a flying animal, but from a pterodactyloid. Mechanics and biomechanics. If the bone belonged to a fossorial animal, if it was a flying animal with being able to sustain very torsion of the bone, for example, and the environment. The environment is more tricky when we study histology of especially dinosaurs, and we start not to realize that if you want to talk about growth and limitations on growth that can explain plasticity, interspecific variation, we need to look to the paleo environment. So studying the paleo environments, the paleoecology, in all their manifestation is really important. I can say this animal probably died, well, he's a lot of growth marks, so he stopped growing time to time for his seasonal. Or there is a lack of food. How do I know that there's a lack of food? I don't know the plants that live at the moment. Right? I don't know what the temperature. I don't know if it's a dry environment, if it's a wet environment. So there is potential and a lots of limitations in paleohistology. So we have several types of tissues that we can study in vertebrates. However, there is some missing here, and they are not here on purpose. And I'll explain a little bit later. For example, osteoderms, they grow in a different way than the rest of the bones. And horns and antlers, because they are not bone derived, they are made essentially of chitin. So chitin is a different thing. So, of course, all the bone embryologically, they start with a cartilage precursor. Is that right, or is that wrong? I think it's right. It's right. Partially right. There is some bones that grow intermembrane. There are the intermembranous bones, especially the flat bones of the skull, mandible, clavicle. They grow directly without the existence of cartilage. So cartilage, anyway, is a precursor of long bones. I would say like 95% of the bone. Osteoderms are also other type of intermembranous bones. They do not have a cartilage precursor. Okay? If you see all of these bones, they have a very flat surface, anatomically more vertical or horizontal, and they grow to the type collagen 1. It's the more dense, while the hyaline cartilage that the normal other bones grow is cartilage type 2. I will not talk about cartilage anymore. Time will be limited, but yeah. So sharks, Zlamobranchi in general, they are made of cartilage. It's rare to fossilize, and also it's not an easy thing. When they fossilize, it's not an easy thing to describe histologically, because there is the permanence of the chondrocytes is completely different from the manifestation of osteocytes, osteons, and other anatomical, macroanatomical components of the bone tissue. So bone, it's a hard material. We have teeth. Yeah, if you go to the field, you paleontologists, especially for vertebrates, like for dinosaurs, you find more teeth than dinosaurs. We say, claim that we have more teeth than dinosaurs, because they are always replacing the teeth. So I wrote a paper like 10 years ago. It was absolutely nonsense, because this here, in the bones, the teeth, there are some marks. He calls them, I forgot. But well, there is some lines that might manifested by these structures, and they were saying that we can do skeletochronology with teeth of dinosaurs. Wrong. You want to bet why? Because they are always replacing the teeth. If they are falling, you can say these teeth, they pop out from the maxilla of the mandible with probably this age. Okay? So, but teeth are very important to give us some aspects of feeding behavior also some aspects of ecology about biomechanics, bite strength, but they have limited information to give skeletal chronology or other aspects of the growth of these of dinosaurs or any other animal. Okay, I think for all of you this is not strange, the composition of teeth, right? So I think I can skip this part and start to go into the bones itself. Of course, canine, it's important tissue also. You have some potentials for the people that study fishes in terms of ontology also. You can see some age-related aspects on the scales. However, the scales does not grow by a position; they grow by accretion. It's more or less like the bivalves. Okay, bivalves grow by accretion, and gonoids you can see the layers, but the layers, the thickening and all the process, the growth itself is completely different and is a completely vascular tissue. Okay, I will now talk also about eggs, but of course eggs are an important component on the biology if you want to understand the biology and reproductive biology of dinosaurs or other amniotes. But it doesn't give us too much information about the animal itself. Now, this is the most important part for you understanding the main actors in bone formation and bone maintenance. So, bone has several aspects during the life cycle of an animal. He has one mineral phase. It's the constitution of hydroxyapatites and other minerals in it. And then there is the non-mineral phase. Non-mineral phase is all related to do with the cellular matrix of the bone. So we have the collagen, and then we have the minerals, and within the mineral structure we have all these agents to make bone growth and maintain. So I talked about this a while ago. This is important to understand that not all the bones have the same origin in embryology, and each of these cells we can see them in histology. So they are a huge source of information for us, and you will see it while in 1 hour and a half at the microscope. So, how many of you are from biology? 3? Only? Okay. So this is not strange for you, right? We give this in biology, or only soft tissues. Okay, so all of these processes are records in the fossil bone. The thing is, sometimes it's not that easy to read that information. So when I'm saying about the bone itself, about the growth of the bone, the maintenance of the bone, and we are talking about also mineralized tissue. So some of these minerals, sometimes they can be completely altered in the sample. For example, if I find a bone in a very dark type of fossilization, I can see perfectly. If it is on a more oxidizing element, sometimes you cannot see even the structure, the organization of the fibrils from the inner structure. So endochondral ossification, this is a classic thing that we cannot see in the fossil record, except if we are very lucky to find baby dinosaurs, in baby dinosaurs. We have some in Portugal, found in Lourinhã, and so basically we find perinates to juveniles individuals. So these processes of the endochondral and the model itself, we cannot see it on the fossil bone, and of course cartilage doesn't preserve. So sometimes we have some void spaces in the dig sites that we can see there is differences on the bone surface. We can say, okay, this was the place where cartilage was, but actually we'll only have the part of the bony tissue, the part of the cartilage with that is the articular cartilage has gone. This is the basic for you. We'll have this, I think, in the classes, how to describe several components of the bone, right?
Not yet.
Not yet, but okay. So, yeah, we'll have enough of this in the future. But it's important for us also to, when we cut a sample, to know exactly where we are in the bone. Normally, what we want when we cut a bone, mainly the femurs, is the mid shaft, and I will explain why later on. Okay, so collagen fibers is something that is a main component of the bone, and also we can see them in the histological sample, as well as sharpest fibers. So sharper fibers are the connective structure that keeps the periosteum, the sensitive part, attached to the cortex of the bone. And we will see in the samples some good examples of this type of fibers. All these structures that you see here, the osteons, endosteum, periosteum, compact bone, cancellous bone, we can see it also in the histological samples. However, they are not very easy to identify. And one thing that, for example, if you, and we'll talk later about osteons as a main component also from bone biology, you have to understand that these osteons, they were not only vascular canals. There are all of sensitive nerves and everything that are inside the canal. So it's not a vascular canal with just a blood supply to the bone. So of course bones, they have to grow longitudinally and transversely. Of course, that's the reason why there is this cartilage structure, so they can grow and be, with time, be substituted by the bone itself. So, bone growth by a position, as I told you before. This scheme here is a wrong scheme. It's a good, it's helpful for us to give an example of what happens. But bones doesn't grow like this, most of them. Some animals do have growth, isometric growth. It means that they maintain the shape of the bone through all his life. Dinosaurs, humans, mammals in general, they change shape during the life cycle. For example, if I found a dinosaur that is a proposal that when they were young, they could be completely fully bipeds, and when they are adults, they can be facultatory quadrupeds. So these have impacts of the bone development and the bone record itself that is maintained in the bone. So you have to think when you find you have quite similar bones, different sizes, but they look different, think this can be ontogenetic. This is allometric growth. And we can see in the fossil record in the bones itself, that's the reason why now we are doing CT scans from all the bones before we get them, not only for having the 3D, a good 3D model for 3D printing or to do any other records. For example, this is from Metoposaurus, from the late Triassic. This is a case where the animal has a isometric growth record. You see the shape in general of the bone, it's maintained during the life cycle. But this is not the rule, okay? So we have been talking about this first. So this is the type of bones that give origin to parts of the skull, to the mandible, to the clavicle, to the ossiderms. Okay. The sampling. Sampling location is really important. Normally, when we want to study animal, we try, of course, to cut the most bones we can, actually. We call it the full skeleton mode. So we want to know how each bone grows. Some bones are good for it, some are not. I told you before, vertebrae don't give you any information. Now, the most valuable ones are definitely the femurs. And the mid-shaft of the femurs, in dinosaurs, for example, there is a specific place where to cut them, normally above the fourth trochanter. Why? Because that process of allometry can obscure the fossil record. So normally, the bed fossil, the record, the growth record is better preserved in the mid-shaft of the femurs. And if we have the homologous bone, in this case the humerus, cut it well too. Because this will give you the balance, because we will show that the one have more growth cycles than the other. It's nothing to do for nothing, just because one is remodeled more than the other, or the other has a development later ontogenetically than the other. This could tell us if it was biped or not, or there is some changes of locomotory behavior, for example. Drill is a good way of doing it, partially good. If you go to a museum, say I will not cut transversely, I will do a drill. You will get better chances to get a permit to do it. It is limited. I tried, and if I don't have a full section of the bone, I don't know the amounts of bone that was remodeled. And we will see this later about the remodeling processes. So cutting is always the best, but we understand that for curators, and specifically when we are talking about holotypes, it's hard to get a permit. So if in between don't have any information and have good information, or have known information, it's better to have a partial information and less destructive to from the bones. This is a scheme of one of the most important aspects of that we can see in histological samples. This is the organization of the collagen fibers, and this plays an important role on the estimation of how fast these animals were growing, and gives also some estimate about their metabolic rate. Okay? But we'll talk this further ahead. So, early ontogenetically, we have this type of bone, woven fibered bone. This is typical from neonates, perinates, very juvenile individuals. And we say matrix, and it's really a matrix because these aspects that we are talking here, they are not related with the vascular system. This is only the organization of the collagen fibers. Type 2, type 1, and type 2. Different bones will have different types of collagen. Normally, very hard, bones that have to sustain lots of loads, like the femurs or humerus. For example, they have the type of collagen is more always collagen type 1 because it's more dense. Other bones have collagen type 2. For example, in our skull, is because he growth intramembraneously, is collagen type 1. It's flex, but is resistant. Okay? So, this is a type of the organization of the collagen fibrils. We can see that organization in the polarized light microscope. Secondary bone. Secondary bone is the bone results from the starting and continuation of the remodeling process. Let's forget now the vascular system. This is the type of bone that normally is deposited when bones start remodeling. After a certain age, we all start have bone remodeling. Okay, and so we have other type of collagen fiber organization. So the secondary bone matrix, when we call it in those stili, this is we definitely assume this is existing only inside the bone, the medullary canal. It borders the bone, the medullary bone is a good indication of ontogenetic status because when we have this, it means that the capillary cavity is stopped enlarged, stopped growing. So then we know if it doesn't exist yet, then we know that we'll start a huge process of secondary remodeling. So this is one of the components that when we talk about the remodeling process. So you have the normal bone that is being deposited during life, and when we start having the secondary remodeling, we start having a vascular, simple, or primary vascular canal, and then we have these lamellar bones or zonal bone that we saw here. This type. It's this is deposited concentrically in layers. It starts like a simple canal with just a border, and with time it gets thicker, and then we have like a cement line. And we know that they are fully mature secondary osteons when they are full of osteocytes. This is an indication also of how fast the animal was growing. For example, we can give you here on the primary tissue, and we say primary tissue because you have the vascularization playing with the cartilage, the collagen. Okay, so we have like a simple canal. Then we have a simple canal, but we can note already there is something happening here. Okay, nothing too extraordinary, but you see for the secondary. So from the simple to the secondary, there's lots of process of bone remodeling and growth. So these tiny things here are the osteocytes, and normally we can see a lot of capillaries. We call them canaliculi. That is, it connects all this remodeled bone, not only for the provision of nutrients, and I have to say that also bone has an important role of cleaning, also to get rid of waste from all the physiological processes. So again, with collagen, playing with collagen and the osteons, you can see different types of fibrillar organization around an osteon. So this is the secondary remodeling, several generations. This starts from the endosteum, around the medullary cavity, to the periostal surface or the external cortex of the bone. Sharpest fibers, it's something that we can see also in the bones. Besides, sometimes you can confuse them with the collagen fibers, but normally they have perpendicular attitude towards the bone. It's completely. I think it's relatively easy to see them, to recognize them. Sometimes it's more easy recognizable than some pathologies. For example, you will see a sample with a pathology today. So this is, you see, these structures that goes oblique to the bone. This is the collagen fibers. Collagen fibers are the things that attach the periosteum, the sensitive part, the area that is the responsible, or is the area that makes bone growth. Okay, so it has to be attached to something, right? Sometimes we have in some areas of the shaft of the bone, we have some structures, similar structures, a bit more bigger. You can see even better. This is muscle attachments, or the migration of the musculature during growth, because there is, of course, musculature attached to the bone, and it migrates with time, and it goes during bone growth. They go getting smaller, and then we have evidences of that growth system. Okay, now here we are talking about the vascularization. Vascularization played with collagen orientation and the type of organization of the collagen. So we saw that there is the woven bone, there is the lamellar bone, but here, when we talk about lamellar zonal bone or fibrillar lamellar bone, we are talking about two playing the same role. So it's the development of the vascular system with the collagen, the organization. So you can see this is several types of organization. This is the interconnection of the vasculars in between them. So we call anastomosis. Is the network of the vascularity between them inside the non-cellular matrix. And you see that we can have several types of orientation. Here you can see here some are more like pits, some have more radial, some are more circumferential. These two informations, organization of the collagen and the vascularity, is the thing that tell us how fast that animal was growing, and sometimes it is a grown animal, adults or not. So this is a type of organization. You see the patterns of the collagen fibers and the pattern of the vascularity. Okay, so you can see lamellar. If the collagen is very, very well packed, very organized, it's a slow growth animal. Okay, but even this slow growth animal can have a lot of algivascular. So it means that it's not growing so slow like that, you know. And then we have, of course, the extremes, the fibrolamellar bone, normally fast growing. If it's a fast growing, so we'll have early on ontogeny, high degree of bone remodeling. And here you have this, the woven bone. It's completely chaotic, but normally has less vascularity, or when has vascularity, it's not that well developed. So this is our science about also the metabolic rates and the growth rates of a certain animal. Okay. So the lamellar zonal bone, the well organized type of bone, not very well vascularized are typical from ectotherms animals. I will not call it cold-blooded animals, but it's typical from these animals. If you go to see a turtle, for example, even a crocodilomorph, you can see that the bone is mainly lamellar zonal type of tissue. It's universal truth? No. We know that, for example, some artificially fed crocodiles, modern crocodiles, they can develop all the type of bone that is a bone and this can give us a glimpse. Sometimes it can be very tricky in terms of how we interpret the biology of an animal. So it's a problem when we have only one species in paleontology. If you have a population, 3, 4, 5 animals, we can see the variation. But we don't, unfortunately, we don't have it. Okay, this is the scheme of this lamellar zonal bone. As you see, there is not many vascular canals. It's very boring, for so to say. So you can see here. You see, this is very well organized. You know, these dots here are the vascular canals. Just a few vascularization. So is an indication of a relatively slow growth pattern. Okay, no coffee. I'll get for another one. Let's go on. So, any doubts so far? Lots, I know. You can ask. It's confusing, right? Do you have any questions from before you forget? The last section, no? Okay. Getting into it again. Fibrolamellar tissue. For everyone that studies dinosaurs, this is the type of bone that we mainly find. He has been generally attributed as a huge. I don't like to have this type of discussion about endothermy in dinosaurs because I think the information that histology gave us is not enough to say that they have a close metabolic rate like a mammal or a bird. High metabolic rates evolve independently 7 times during the evolution of animals. If you think, like for example, a huge sauropod, So, does that animal needs to be endothermic?
I think so.
Yeah, why?
Just being an ectothermal could not reach its size and be so active as the fossil evidence shows.
Sauropods. Animals with 8, 10 tons.
They might have trouble managing their heat.
Exactly. It's a huge mass. Every time that animal walks, it releases a lot of energy. So the perspective of these animals is how get they reach the heat they produce by just existing. Okay? And they grow hyperly fast. Does the bone tissues are typical from a mammal? No. A bird? No. Okay, but the growth rate is different. So normally the question of the metabolism is much more complex than being ectotherm or endotherm. Okay, so we can say that at some point, if the sauropods were metabolic, or we can discuss at that level about the eventually the resting metabolic rate of an animal. It means that the amount of energy that one animal consumes while quiet. Okay, but a huge sauropod, he has a source of heat by itself. Okay, but he doesn't have the type of bone tissue typical from a mammal, because the life cycle, the growth rates, and the remodeling of these animals is completely different from a mammal. A mammal has a sustained growth. It means that the type of bone that it deposits during life is always the same. Doesn't change. In dinosaurs, they can put femoral bone in one moment, and suddenly it's another type or a variation of that type of bone. And they have like growth marks or lines of arrested growth. We will talk about it later. Wild mammals, well, they have it in a different expression. It's not a cellular like in dinosaurs. But this is the fibrolamellar complex. So this is, you have like, this is the lamellar bone, woven bone, lamellar bone, woven bone. This is a sauropod. Okay? And we are talking bone, not tissue. We are talking what is inside here. So the mix of the fibrils is completely odd. Maybe it's a result of giantism. And Professor Otávio has studied with Professor Sander one dwarf sauropod. He grew relatively fast, but he was small. He was a dwarf. He gets full adult, and the histology, even from these animals, is completely different from any other animal. So this is the model for basically the structures, that indications that this is a very fast growing animal. You see these open spaces here, this is vascularity. So he anastomosis a lot. Is fully irrigated with blood vessels. Okay? And this only happens when an animal grows fast. So more organization, slow growth. Chaos, crazy structures, fast growing process. So I will not talk about here from the plexiform type of bone and that, because it says is highly controversial for many authors, but this is like you have a mix of 2 or 3 things all together, and no one agrees with this type of designations. So I will not talk about it For now, even for me, I don't care. I just follow one author and that's it. There is now the next International Congress. This will be discussed because we need to create some uniformity on the nomenclature because everything is chaotic. If I'm reading a paper from one guy that told me about the plexiform bone, that is another has another way of saying it. It's a mess. So this is now for you to see like types of vascularization. Again, this vascularization, what we call the woven bone, femorolamellar bone, this has to do with the type of organization of the collagen and the type of vascularity. But in this case, you can see, forget the vascular system, but you have these well-organized structures. Slow growth, fast growth. But fast growth after the perinate stage or ontogenetic stage, because then you can have the woven bone. The woven bone, actually, it was the opposite. Sorry, woven bone. It's typical from very young animals. So again, several types of organization, eversion or secondary remodeling.
Bruno, can you explain, so while you're doing this, can you explain exactly what to see in the pictures so they can understand? Because you have pictures and the results, and it's cool to understand exactly
You
what
want
it
to
is.
see this one or? Yes.
No, in general, in general.
Okay, so, what I'm saying is about these, you see these packets, very well organized packets here, layers, small layers, this is collagen. This is vascular canals. This here, resorption lines, we have no secondary remodeling, no osteons. Here you have the fibers. They are not well organized, they are in chaos, because what you see here, these voids, is the anastomosis, the blood vessels. It's blood vessels everywhere. Okay? And then you have here, this is resorption cavities, but what you see is primary canals. And this means that this animal grows very fast. It's a very young type of tissue. I will show you some examples with better images from Portuguese dinosaurs. Again, you have the secondary remodeling. You see they are, you can see these structures. Okay? Here is the vascular canal. And this is the lamellar bone that starts to deposit around this canal. And you can see they are almost pumping on top of each other. It looks like they are. So we can say that there is at least 2 generations. You can see 2 generations of remodeling. And we can have more. Okay, if you see, for example, another one that is eating, this one, look, this one, or rather this one, eats, sort of saying, the one that was there previously. You see? Because it should be completely round, but it's cut it here by this. Okay? And then you have the small dots. You see? This is osteocytes, bone maintaining cells. When you have a lot of osteocytes, there's also an indication of high growth rates. I think I have a better
So, may I make them a question to see if they... Imagine you see this in an animal, in a bone. What is this? Each one of them is an osteon, right? Is this a juvenile or an adult? And why? Tentatively. Trendly. There's a lot of tricks about this. So these osteons were deposited first. These are secondary osteons that have eaten this part. The osteoclasts destroy the bone and replace the bone here. So to do this, it can't be a neonate. It has to be an individual that already deposit the osteocytes and already destroy the part of the bone and redeposit on secondary osteons. So secondary remodeling and secondary osteons normally tends to indicate an adult more towards a maturity. Let's put it this way.
Yeah, it's always tricky, and it is one of the things that there is not many dinosaur samples histologically, but yes, this is a sign that something has happened to the skeleton of the animal. Normally, we start to have secondary remodeling, first generation, second generation, third generation, when the medullary cavity starts to expand. This is one first indication of maturity. I would say, from what I saw from what I've been studying in other dinosaurs, it occurs normally when they are juvenile or subadults. And then there were other things that play a role on it, is also the growth marks. So this is the difference between dinosaurs or some archosaurs and mammals or even birds, because mammals don't have this type of growth cycles like the mammals. So there is some modulations during that we can see in the cortex of the bone. By the cortex, I say the exterior surface and the medullary frontier, the endosteum. Okay. So with time, we see that there is changes on the type of the bone that is deposited. It means the woven bone or the fibrolammellar bone. Again, when I say fibrolammellar bone, it's chaotic arrangements of collagen fibers with high vascularity. Okay? So matrix has one thing, bone tissue is two things: vascularity and organization of the fibrils. And you can see this on the modulation, on the alternation of that you can see in between the cycles. And this is a schematic. So you have the endosteal bone. When you start to have secondary remodeling, you have a deposition on the medullary cavity. Not true that it happens secondary remodeling. I see in my samples I have just 2 or 3 secondary osteons just after the lamellar type of bone tissue on the medullary, around the medullary cavity. So this is what we call growth marks or growth cycles. I don't like that much of lines of arrested growth, to be honest. Mainly because we don't know if the bone stops growing. The skeleton that bone stops growing from some time, but I don't know what happens in the rest of the body. As I told you, if I probably will cut a humerus, so at the same age of development, the femur can have a line of growth, or a line of arrest growth, but not the humerus. Okay, so it's tricky to. This is complex during skeletochronology in dinosaurs based only on this type of marks. I'm always very, very careful about it. That's a reason that I prefer to say it's juvenile or it's adults or it's sub-adults than counting age. Okay, I think it's the maturity of the skeleton that has to account. But okay, so this is the type of growth marks. I will show you better. But then you have this good scheme, and you can see the type of collagen fibers. Okay, this is collagen. Secondary remodeling, endosteal lamellar bone here, and then you have type of circumferential anastomosis of the vascular canals in the type of laminar bone, and we have the radial distribution of the vascular canals. Okay, all this system is what you can see in the fibrolamellar bone. In the lamellar bone, you don't have this variation. It's practically all the same. Basically, is this. But this type, we can say this animal here, he starts growing relatively fast. He starts, then he moderates the growth, and suddenly he has a bone spurt. Not only because of thickness, but by the type of the organization of the vascular canals.
Bruno, could you actually see a bone like this, or this is the skin?
No, this is the skin. No, this is the skin. No, in fibrolamellar bone, you don't see this. This is
so that's actually impossible, or just
this
a
and
chance?
this and this. All this you can see in the fibrolamellar bone. Okay. Here, why? This is not real. Because it's simple canals. Okay. I know why they did it. In the skin. It's just a simple to see ontogenetically some remain of primary bone. Yeah, maybe it's not the best scheme actually, but you can give. So when you see articular bone, radial bone, laminar bone, this has to do with the vascularization. Okay. In the complex of fibrolamellar bone, you can find these three. Okay, in the fibrolamellar bone, you can find, of course, simple canals or primary canals, of course, in the periosteal surface. But this is completely wrong, unless they want to draw some resorption lines. But they should be more or less in this position. Okay, so this is basically an animals and the areas of arrest growth. Normally, you have the bones growing normally, and suddenly you have a deposition of very well organized lamellar bone, avascular, acellular. It means that it doesn't have any osteons or vascular canals in it, nor they have osteocytes, nor they have canality. Okay, it looks like almost the same principle of cartilage growth, because cartilage is a cellular in that sense. So this is the way of vascular canals connect to each other. This is a good scheme. Why not? Green lettuce. Green is always good for the eyes, at least to wake up it. So you can have a type of chaotic type of bone. You see, there is some here. You don't have a here. You have like a radial connection in between the vascular canals, but in this area it's much more chaotic. So this is a type of animal that is growing really, really fast. And you see there is an endosteal line. It means that the medullary cavity stopped expanding. And then you have this cellular. You see there is no osteum, no cells here. As you can see, This is anastomosis, of course. It's normal they go through all the bone, but there is no cellular activity on this position of that bone. You can see any? I think you can see any osteon or something in this line? No, there is none. So same here. This is in Myosaur. Chaotic bone, resorption, cavities, vascular canals going everywhere chaotically. Stop. Starts growing. Here we can see these probably are some radial vascular canals.
Which side is external and internal?
The periosteum should be this side. Okay, so this is the periosteum. If I turn it, we are more or less in this position here. You see, endosteal, very laminar, organized bone. Then mature or secondary osteals. Okay, so there is one element that is really important for establishing how adults this animal, this particular animal or bone. If you are with individual bone, you can say this bone growth like this. If you have something that is called external fundamental system, you can say this is an adult animal because it stopped growing. I will show you some pictures. This is, you have bone growing like this, okay, fibral and bone. And when you arrive to the outermost cortex, you have this very well organized packets of bone that is, you see in the lines, that is the distance between them is getting closer and closer and closer and closer. And then you see that the amount of vascularity reduces absolutely. Organization of the fibrillar the cartilage is much more well organized. So we can say this is a fully adult individual. It doesn't mean that he's senescent, that he's old, but you can see that. You'll see in the sample what I'm saying. One of all the things that we can see on the bones is when, imagine that you have this life cycle. Okay? So imagine somewhere here you start having the lines more close together, okay? From this point. But you still have some vascularity. But from this point out, the bone starts to, the growth marks start to be closely related. We interpret this transition as when the animal is sexually mature. It doesn't mean that he is skeletally mature. So he has reached the sexual maturity, not skeletal maturity, asymptotic size. So if you have this pattern, then we know that he's slowing down the growth, he's getting adults. You reproduce, and you can live happily for some more time. Probably will die. But the truth is, we don't know many dinosaurs that are full adults and senescent. It means that they are really old. Maybe draconics, it looks like, but let's see, because we have to think that the growth rates of the death rates of these animals are quite huge. So one of to me the best way of, but is the most difficult one to get, for example, to know the skeletal maturity or the even allometric growth processes. What change exactly in the shape of the bone during ontogeny, and to have quite sure if the lines of growth they are correspond truly to how many years this animal lived. This is possible when you have populations of animals, or at least you know that it's the same species on different ontogenetic stages of growth. So basically, you draw the contour, you draw the growth marks, and then from the different animals from these different class sizes, and then you overimpose them, and you can have more or less an idea. But this results if you have like the individual until the more older than you can get. This is difficult, it's hard, and because there is not many baby dinosaurs, and there is not many baby dinosaurs with older individual or juvenile or individuals or subadult individuals or skeletal mature individual, non-skeletal mature individual. So it's problematic. Normally I count the age by the median, never by the top, because I don't know exactly how much was remodeled, and I'm not seeing because it was remodeled. This evidence, this first bone that was deposited on the early stages of life, is not present there anymore. It was absorbed by the expansion of the capillary, the medullary cavity. Okay, so it's tricky. Other method.
Can you explain it again briefly? The calculation.
This is there is other things. It's retrocalculation, but we will talk about other model.
Okay.
This one, it's for example, Vivi and Lee. They published this paper. They got several Allosaurus femurs. They cut them. They have small individual, big individuals. That's one problem. They are not all from the same site, the same formation even. So it's complicated. And then we will see this with our Iliosaurus, because populations is a problem. And what they did is from the smaller individuals to the grown one, they cut, they saw, they counted the growth marks, they draw them, and then they overimpose the draws. This is over imposition of the lines of I think was 8 or 9 individuals. It's not bad. Okay. So this is estimation of age for each. We say, for example, here is 0 age. Yeah, could be. Why not? The problem is always when did he grow? When did he born? It was 1 year ago. So if there was 1 year ago or 2 years ago, there is already a lot of resorption. And these animals, they grow relatively fast. So if they grow fast, it means that the remodeling, because the bone is eroded from the inner cavities to be deposited on the outermost part in the periostal area of the bone. So, okay, so ground curves is another method because basically established by Martin Sander and Nicole Klein, taking in account the giantism of sauropods. They create this model and also another 2 models of growth to explain growth or to describe growth in dinosaurs. For example, we can again try to recalculate the tissue that was lost in ontology, that was restored. In this case, for example, what they did is they go by the type of tissue that they associate with different types of tissues from different animals from the same species, And basically they make a calculation of how much bone is present that you can see until the animal dies. Okay, so and they have like a linear curve, but this goes from the principle that the growth rate is exactly the same for all the animals, which is not necessarily true. Okay, and there is another problem to me with this model, is that the bones are not isometric. So if you count, if there is remodeling from the back of the femur, because he's reshaping and put more bone in the front, it doesn't mean how do you measure it, because it's not a complete circle. So it's not from my point of view, it's not the best one. And, to be honest, there is not many people using this process. Now, what has been done, and I want to apply this for ornithopods, is the ontogenetic, histological ontogenetic stages. So this is much more accurate because you don't create any fantasy. It's what is there, it's there. Okay? So, and then the funny thing with this method is you can do it with histological, the thin section, or you can do it with polished sections. For sauropods, not for other animals. So basically, what Sander and Nicole Klein they have done is, and they could find this pattern. For example, Titanosauriforms, they have this pattern of bone remodeling and the alternation of the type of tissue. This is valid at least at family level. And for example, Camarasaurids was exactly the same result. So instead of saying this animal died with this age, it doesn't matter. We say that this type of tissue that he is depositing now, it's more or less already mature or a few mature type of tissue. Because, yeah, as I told you, say this guy died with 15 years. Could be, could not be. It's preferable in terms of skeletal chronology, talking about juveniles, matures, or full adults, senescent individuals. Okay. Secondary adaptations in bone microstructure. So we come all from the ocean, the water environment, right? So our ancestors, they have a certain type of bone tissue, right? Which one you think it is? This, right? Okay, that's the reason why we call it secondary adaptation in bone microstructure, because then we change more or less for this. Flying is relatively convergent with between pterodactyloids and birds. But yeah, this is one of the things that when you see a histological sample, you can say more or less a type of ecological or environment or what that animal was. It was fossorial, it flies, it lives on land. And we can say if he lives in the deep ocean freely, or is more related to coast shore lines. So you see that in these cases you have a completely full medullary cavity, you have a relatively reduced one. This one is much more dense cortical bone, and then you have different type of adaptations of animals to their environments. So this is birds. Why? Yeah, because they need to fly, they have to be light, the bones have to be ready for more torsion than compression, for example. If we look, this is one of the things that you can see in pterodactyloids, flying reptiles, for example, is this type of convergence. The bones, they follow the same principle. The growth, the growth strategy, or even reproductive strategy could be different, but this is completely convergent because this is the best way or more successful way to accomplish their lifestyle. Yes?
I have a question, but
Yeah.
that was the bone of a dodo, it says there, so that's not a flying bird, but it still maintains that type of structure?
Yeah.
Okay.
It's amazing, yeah. Because they stop fly evolutionarily, not.
Yeah.
So, but what I need to say is the type of tissue, for example, is already from a fast-growing. You see, these structures.
So you could assume that evolutionarily the dodo stopped flying recently, so the bones haven't.
Probably
modified
yes.
that much,
Yes,
so-
but it's completely hollow. It's not typical from a terrestrial animal. So this is the thing that we have to pay attention where we are talking about one animal. The same I will show you, for example, for okay, this is the types of bending mechanisms of the biomechanics of bone, and I will talk about, for example, penguins is a good example of a bird that is there's no bones. Why? Okay, this is a flying reptile. Okay, fast growing. This one is something that we call aging. This individual was skeleton mature. There is no cellular bone anymore. The last growth moment was this one, and I have this here. So you see, in some things bone doesn't lie. Of course, we have, like, for example, lagomorphs. Lagomorphs have some bones that are the bone wall are relatively thin. Okay, but this is to make, because they have to be fast running. But not all the limb bones are like this. Normally, the tibias and the fibulas are dense, for example. There is two ways of when you talk about the secondary adaptation to aquatic environment. Is we have one non-pathological condition that is osteoporotic bone, is the loss of density, but it doesn't create hollow structures. And the paleostotic type of bone is the thickness of the bone. They exist because these animals have different ways of diving and living on water. For example, this one, it allows them to control diving in shallow environments. If you have this type of bone, doesn't resemble you guys any type of bone from any animal. No? Dolphins. Then you have the manatees. Very slow animals. They go like slowly. The bones are completely dense, but spongy inside. This is, for example, whales are completely spongy, but this question about the structure of the bones of the whales, in my interpretation, is not only related with diving capacity. I think the bone is also designed to hold huge pressures. Sperm whales, I think they can dive like 2 kilometers, something like that.
They dive to the point where there's no light.
Sorry?
They dive deep to search for
Squids for food, yeah.
So probably to the point where there's no light visible. So very
Okay.
300 meters.
But 2 kilometers down is a huge amount of pressure for an animal with a huge mass. Okay, so I think the microstructure of the whales is more or less in between this and let's say this maybe. The ichthyosaurs is more like the dolphins that we see before. But I think in the whales it's if you see that the type of architecture is something that allows to the weight to disperse equally in the bone itself, not to be crushed by the high pressures. Okay. Now this is the same different animals that lived in different times, different environments. They have more or less the same architecture of the bone tissue. We can hopefully this software is very good. That's the reason why I put it here. If you want to, especially for aquatic animals, we cannot. It's not big results for terrestrial animals, but this software is really good because it has some algorithms that allows you by the density to see if they are more correlated to, for example, the kind of microstructure from a dolphin on an ichthyosaur than a turtle whatsoever. Okay, because there is a good index of porosity in the software itself, and you say if you have these results, so you can say that this is more the type of environment for even an actual animal. This could be the type of environments it can live in. So, the porosity of the microstructure, I was a bit ahead about talking about the whales, but is a good example. When people talk about whales, oh, this is very similar because it's very porous. The porosity in whales, I think, has a second reason to be, and it is not only related with the living in the open ocean. It's because and how they dive on deep. They can dive, and everything on that animal, it's well planned to get in that. Now, almost ending. This is an example from dinosaurs from Portugal. I studied three specimens from Briosaurus from Portugal, all attributed to Briosaurus. Now I have 12 samples overall. It's good, not a population, but we're getting closer. So this one is a Gen 4, is this size. Is the same size as the supposedly adult, but it's not adult, is the biggest Dilophosaurus that is known. For if you don't never heard about it, Dilophosaurus is a very well known ornithopod dinosaur from the Tendaguru beds from Tanzania, and they were found 100 and something specimens in one layer. So Hubner is a lucky guy. Studied histologically practically almost of them. So the biggest one has the same size as our femur. This one is the holotype of Eosaurus, is like this, the femur. Okay? And this one, it's more or less the same size, like I think it's less 1 centimeter. So, what to expect? The big one I will have finally an adult, a fully adult Dinosaur, because in the fossil record there is not even one adult, fully adult Dinosaur known. They all die before reach skeletal maturity. So, yeah, I think for the first time it makes sense, because if I have these individuals, we consider these individuals like sub-adults, or juvenile sub-adults, makes sense. So this morph could be the one. So yeah, so I make one transverse section from this one, and the other two were drilled. So this is from the holotype. So after you saw in the class, now what is this? No one guess? Lamellar bone deposited on the dostal part of the capillary cavity. Okay, so you have this is well organized or badly organized type of bone.
I think it is well organized.
Is
Which one?
is well organized. Look here.
It seems to have different areas of organization.
Yes, of course, because this is fibral and not bone, and you are not seeing the anthophytes, but you can see the fibrils are running fast, and you have so many vascular canals. Okay, you see, you have a lots of vascular canals around here, few here. You can see here, you can see here. I'll talk about this in a while. But yeah, and you can see that there is organization and vascularity. Two things, okay? Because you have circumferential vascular canals and you have radial vascular canals. Only fibral and bone is this, fast growing or relatively moderate to fast growing. Because yes, we have some tabular structures, yeah, it's not like the big group. But you can see this is slow growth bone. This is a little bit more slow growth bone. This is from the femur from the other animal. You see, resorption cavity. It's relative. You can see here. It's relatively porous bone. Lots of bone cells everywhere. We have the vascular canals. There is small dots that are the osteocytes. And this animal, if I go by the skeletal chronology, by the counting on lines, this animal and this animal, they died. It's 5 to 6 years old. But we can see by the cycle, it's that we have something happen here, and here and here. These lines are more packed, closely packed, aren't they? So you have growth. This is missing cycle of growth, remodeling, and then suddenly you have here, and then they start to have big packets. So this means that this animal, he has reached a sexual maturity. The same for this one. Actually, there is one line there. You see these two lines?
These lines are the legs, right?
Sorry?
Are legs.
Yeah, yeah. They are stacked again together. Sexual maturity attained. Head throw calculation, 5 years. And then you have from the biggest one. So the biggest one, crazy. because this is chaotic, absolutely chaotic. You have practically no stop of growing. You have modulations, different type of arrangements that you can see. But this is huge, fast growing animal. And when I said, yeah, probably 7, 8, 9 years maximum. Sexual maturity attaining, but no signs of still stop growing, because in the other ones you have well organized type of tissue, and this is the smaller ones. Here, yeah, it's not that fast. Here, absolutely chaos. No signs of stopping. So, this type of tissue is what we can say when individuals are fast-growing young animals. So the bigger one is the tissue types, collagen organization, orientation of vascularity, amount of vascularity, amount of osteocytes, amount of canonically to irrigate the bone. All types of bone of a very young animal. So what? And now? Remodeling. The others have no remodeling. Just one this femur. Actually, I don't have any close by picture. Yeah, here there is a small two secondary osteons, and that's it. This one is absolutely none, and then I was expecting not to have. This is a small amount that I can see. This is a break from the collapsing of the bone. No remodeling. Do you see any remodeling? You don't see any secondary remodeling. No? I don't. I'm very bad teacher. It's so evident. They are all here.
That's all secondary.
This is secondary vascularity. This is a close by from the, but this is a tricky thing. So we talk about secondary remodeling according to aging. But secondary remodeling occurs also on pathological conditions, on bone repairing. So this animal had an injury, and this remodeling is the bone regrowth. Okay? First time pathology of a Portuguese dinosaur bone. Initially I thought that it could be sometimes, but it happens sometimes when you have strong musculatures getting in the bone. You can have secondary remodeling when the animal are growing. However, they are really followed by huge amount of sharpest fibers, and it's not the case. But you see, this is the periosteal surface. You can see there is still bone growing in this area. So this animal, he had attained also sexual maturity. For my accounts, with 7 years, but is still actively growing like crazy, and has 3 times the mass from the other ones that have like more older type of tissue, more mature type of tissue.
But that big amount of vascularization, is that because of the healing or because
No, because the healing is just a bit, it's just this part. You have a circular bone, it's only this part.
Yes.
The rest of the bone is perfectly normal. So you should expect to see this here.
It's very vascularized.
It's crazy. This is fast. So the thing is, it's another species or it's a different population. The thing is, if I want to name another species, I can say that I will invent here some crazy characters. But no, the femur is exactly exactly the same as the others. I cannot say morphologically that there is any difference. And we did geometric morphometric to see any variation. Nothing comes out. So my interpretation at this point, unless we find more from this animal, it's that we have in the Upper Jurassic of Portugal 2 different populations of the same species, separated in time and geographically.
Do you think it could be males and females that having really different strategies, growth strategies?
Dimorphism, it's complicated, yeah. With histology, dimorphism, if I have, I didn't talk about it on purpose, not to not create even more confusion in your heads about histology. I know that this is a nightmare. But there is something called medullary bone. This is bone that is formed inside the femur. It serves in the females as a reserve of minerals for in the legs, the eggs. Unless I find this, it's difficult to say if she is a male or female. But I think if it's true, I cannot put that apart, okay? But it's not the most parsimonious.
No, but some mammals do it, having so much of dimorphism on growth, in which either females or males are way much smaller. So I wonder if you could see such a striking differences between two kinds of bones that is just sexual.
No, but they share some characteristics. For example, in the radial anastomosis, they share circumferential and the radial anastomosis of the vascular canal. But the growth, the organization, this is what you expect for fast-growing young juvenile. I don't see to see sexually dimorphic. I should know if it is adults. For example, and it is they attain sexual maturity at last. I cannot see, and then Martin Sander also we have been discussing this for the last 2 or 3 years. These specimens, because this is one of the aberrant things, and we have to follow the rule. The most parsimonious answer is probably the most correct one. Now I will sample more. I have now in total, I think it's 12 specimens. Okay, femurs on different ontogenetic states. Maybe I can see some difference there. This specimen, it's a pity because the femur is this one. This is a part of the femur. It's completely collapsed here. But I will rebuild this digitally. I can do it. Didn't have just the time. And this is a drill. It's a problem of the drill. You have only this. So I have to resample. And so the problem is I will have to do the sample exactly where I did it before, but then I will have a void that I have to digitally fit this in.
So this is a core, a drill.
This is a core, yeah, the only thin section that's already produced from not very well preserved. And then this is the case of the sample is not the best one because of the thickness of the thin section. It's not the best. But yeah, when you start getting in and the problematics, then you can see some interesting things. And yeah, actually the femur, this is the tibia, actually, this is a tibia, and the tibia has a better record than the femur. It's not strange. It happens all the time. Sometimes you have, and in several dinosaurs this happens. The tibia has a different record from the femur. I think this has to be with the height rates of remodeling on the femur, because the femur gets much more remodeling earlier than the femur. The femur is an hypothesis. So yeah, so some of you were already sleeping.
Thank you so much. We have some practical exercises, right?
Yeah.
Fantastic. Let me just stop recording.